Okay, well, thank you firstly so much to the organizers for inviting me. It's really a lovely pleasure to be here, and also it's great to see the fantastic lineup of speakers that you've brought over. So it's really been an enjoyable day. Thank you very much. So for my talk today, I want to really pose one big question, which we're really fascinated by in my lab, which is, can mutations in tumor genomes can mutations in a genome give rise to a tumor via a long non-coding RNA? And so I'm going to return a couple of times to this image of a crashed aeroplane as an analogy for this. When a plane crashes because of some defect, during the process of crashing, it also accumulates a lot of damage. And the objective of the people who investigate the crash is to sort amongst all of this damage, which was the damage which actually caused the crash in the first place. And in the same way, those of us who are studying tumor genomes, although tumors contain many lesions and many mutations in their genome, we want to find the mutations which actually caused the tumor in the first place. So just to give a bit of background about my lab, so we're located at the University of Bern, here in the capital of Switzerland, and we're funded by this great project called National Centers for Competence in Research RNA and Disease, which is a network of up to actually 40 research groups now in Switzerland. And at the end of the year, I'll actually be moving back to my hometown in Dublin. And it's actually very misleading, this photograph, because it's never as sunny as it appears to be here. I think they've used Photoshop in that photograph. And my lab is mainly focused on long non-coding RNAs, or lincRNAs as I'll call them from now on, and we're interested in cancer, in cardiovascular disease. Our main tools is either bioinformatics or CRISPR-Cas9, although I won't talk about that so much today. And we're also interested, similar to Igor, in this question, of course, of what is the sequence function code of lincRNAs. But today I'll focus more on our cancer work. So for simplicity, I'll just remind you that although there's a lot of debate about what the modality and mechanisms for regulatory lincRNAs are, we will please assume for the purpose of my talk that lincRNAs that we're interested in are acting as a mature RNA molecule as shown here in blue, and possibly interacting with other biomolecules in the cell to affect some kind of regulatory functions. And of course, lincRNAs have been gathering growing attention in the field of cancer biology. We now have growing numbers of examples of lincRNAs which play oncogenic roles, for example, by promoting tumorigenic hallmarks or opposing tumor suppressor hallmarks. And inversely, we're also aware of large numbers of tumor suppressor lincRNAs which play opposing roles to those. And we've growing numbers of publications, including from many of the presenters today, in very accepted roles for lincRNAs in tumorigenesis. And this is leading to quite some hope that we could target lincRNAs therapeutically, such as in this seminal paper by Sven Diedrichs' lab, where we can introduce, for example, moieties like antisense oligonucleotides to inhibit lincRNAs. And at least in these pilot studies, they give quite favorable responses in terms of tumor growth and metastasis. So taking a step back, the world of oncology today, obviously we fall very far short of where we would like to be in terms of being able to treat patients with cancer. I think we can summarize this into three key problems with the present world of small molecule-driven drug discovery. Firstly, many of the driver mutated proteins which are causing tumors, many of these remain undrugged, so there are no small molecules capable of targeting these mutated proteins. Secondly, it's unfortunate that many of these mutated proteins are core signaling molecules and which are expressed rather widely throughout the body, and if you block them, you get very devastating side effects for the patients. And third, and possibly most important, is that even when you develop a very potent and effective small molecule inhibitor, because the tumor is an evolutionary process, it rapidly evolves resistance to this new evolutionary pressure you've introduced, and typically the patients will relapse in a matter of months or years. Okay, so we have not solved really any of these problems so far, and I think there's at least some hope that we could address these issues using lincRNA therapies. Firstly, we can target lincRNAs with these antisense oligonucleotides, which I told you about, which we can program and develop very rapidly in the laboratory, and therefore hopefully economize and shorten the drug development pipeline. Secondly, I'm actually showing an example of Samson here, but many link RNAs are very specifically expressed in tumor cells, and therefore we might avoid this problem of side effects. And thirdly, we might be able to combine these link RNA oligonucleotides with existing therapies to try to avoid this problem of drug resistance. So actually, Anton and me are also using Google to do our image searches, because I was happy to see we both have the same iceberg and we're telling the same message. But what I want to tell you here is that the other attractive thing about link RNAs is we have this massive space in which to search for novel drug targets. And so just to remind you, we have about 1,000 link RNAs where we have any kind of functional data to date, and present catalogs are moving upwards of 50,000, some of them even reach 100,000 unique gene loci at the moment. So this is a really massive area in which to search for new drug targets, but this also introduces a massive challenge of technically how can we screen such a large number of potential candidate genes for drug targets. And so this is what we've been thinking about in the last few years, how to attack this problem. Of course, the classical way, which has been very effective, but the classical way to find link RNAs involved in your cancer of interest is simply to do some kind of gene expression analysis between tumors and normal samples. You find differentially expressed genes and you start to knock them down one by one till you find what you're looking for. This has been effective, but it makes some very key assumptions. It assumes that changes in gene expression reflect function, and it also assumes that the cells in the bulk of your tumor are really reflecting what's going on in the most important cells, which are either the stem cells which are originating the tumor, or the original cell many years before, which actually got mutated to give rise to the tumor in the first place. And technically what that requires, of course, is just RNA sequencing or microarrays, which we've had for a couple of decades now. The second approach, which is gaining much more prevalence now, is a functional screening, where we use either RNA interference or CRISPR to inhibit many link RNAs or protein coding genes in parallel and look for a functional readout. This is also very effective. What's nice is you get a direct readout of function, but you still must do these experiments, or usually must do these experiments in cultured cell lines, which many people acknowledge are not a great, really a great model of cancer. So that's been done for about 10 or 15 years now. And what I want to show you is a kind of a different philosophy where we try to look at a genetic approach for discovering new cancer-causing link RNAs and try to go back to the original cells which are giving rise to a tumor and ask what mutations are occurring in those cells and which genes they're occurring in. And what I'm going to show you later on is that the reason that nobody has really done this so far is a very simple technical reason, is that we have not had access to the data which you need to do this type of analysis, and that data is entire human tumor genomes and maps of the mutations which they carry. So first I'm going to show you the work of Adrienne for the next section. So what I'm going to tell you about in the next 30 minutes is firstly how we've been using mutations in mouse models to discover putative cancer driver link RNAs. Then I'll move on to tell you about how we've been using mutation data from tumor genomes to answer the same question. And then finally, I want to finish up with some recent experimental evidence we have to really ask this question directly of whether mutations in link RNAs can promote tumorigenesis. So we came to this because a few years ago, actually, the paper that Anton and I showed you earlier, we were manually collecting data sets of link RNAs driving or involved in cancer as a benchmark. And when you start doing something manually and you realize how long it takes you, the first thing on your mind is how to speed this up and make it automatic. And that's when we discovered this whole world of transposon-based insertional mutagenesis, which people have been doing for years, where to me it's a very elegant approach where you develop some kind of piggyback transposon which contains a cassette which, when inserted into the genome, when inserted inside a gene, either blocks transcription of that gene or else upregulates transcription. That's depending on which orientation it falls in. So normally, when these insertions, when done randomly through the genome, has no effect, but occasionally this insert will fall within an oncogene or a tumor suppressor, and when you inject a population of these cells into a mouse, and then you retrieve back the tumors, you can sequence the inserts and discover where these inserts have fallen. So this is a very widely used and effective way of screening for new oncogenes and tumor suppressors. So all these people have been doing this for years, just focusing on protein coding genes, but we realized you could also use these data sets to try to discover oncogene or tumor suppressor link RNAs. So this is what Adrienne has been doing. She developed a really rather simple pipeline, which I show you on the left, where she takes the entire data set of known TIM insertion mutagenesis locations. She takes the entire set of these, which are in mouse, 26,000 of them, and she simply maps them back to the human genome and asks in which genes these things fall. So here's an example where we have, in mouse, this link RNA on the forward strand and an insertion site shown here by the lightning, a couple of kilobases upstream. And here, when you map this site into the human genome, you find it's falling now in the orthologous link RNA just inside the first intron. So does this work? Does it tell us anything useful? So what I'm showing you here is the number of insertions in human normalized per megabase of gene sequence. And here in red, I'm showing this is protein coding genes now. These are protein coding genes which are known previously implicated in cancer, and these are all other protein coding genes. And see, what's very nice is compared to intergenic space, where we expect where there's no genes, no annotated genes, we see a huge enrichment from here to here in known protein coding cancer genes, just as we expect. So there's an enrichment, and it's enriched also over what we expect for protein coding genes in general. So how does it look for link RNAs? Now, if we look for link RNAs, we see overall a lower density of these insertions, but these are still far higher than what you expect by random chance. So what's quite nice is that when we take this manually curated set of cancer-related link RNAs from the literature, they are much higher enriched over background, but we also have a large population of novel link RNAs which carry these insertions. So this on its own is described in more detail in this publication which came out, this Peacock companion paper. But what it's telling us, I think, almost for the first time, is that we have a large amount of functional conservation in these link RNAs between two species. So Igor talked a lot about finding orthologous link RNAs between species at the gene level, but this is to some extent taking a step forward and asking, is there functional conservation between those orthologous link RNAs between human and mouse? And also, so these are the known, this is 21 previously described cancer-related link RNAs, but also here we now have 102 link RNAs, annotated link RNAs, which have not previously been associated with cancer. And so these we are now adding to these manually curated sets of cancer link RNAs, which are going to get published quite soon. So, of course, these are all just predictions. We have to see if they have any predictive value. So coming back to this example, which I just showed you, this link 570 was previously described by Ramin Shikatar's lab as ncRNA activator 5, but it was not previously associated with growth phenotypes or with cancer. And now what Noria did is she designed 3 different antisense oligonucleotides and also 2 CRISPR-I constructs, which I'm not showing you, but give the same effect. And then she introduced these into HeLa cells. We chose HeLa because actually they were the only cells we could find which express this link RNA. And you see this is the growth, the subsequent growth of the cells. You see for all 3 of those antisense oligonucleotides, you get a reduction in growth. So at least in one case, supporting that this orthology method can identify true cancer-related link RNAs in human cells. So let's move on. So that was the mouse work. Now let's move on to the human identification of non-coding drivers. And this is the thesis work of Andres, who just graduated. And let's just take a step back and review what we know about tumor evolution. So the standard thinking on how tumors originate and evolve is that this is essentially similar to a Darwinian evolutionary selection process, where we have normal cells in our body which are bombarded constantly by random mutations. Most of them are repaired, but occasionally the cell allows mutations to remain. Most of these mutations are phenotypically neutral, so they do not affect the behavior of the cell. These are described as passenger mutations, which are shown in blue. And occasionally, when you're very unlucky, you get a mutation which actually alters the phenotype of the cell and gives it a growth advantage over its peers. And this is what's called a driver mutation. So because driver mutations give a growth advantage, they come under positive evolutionary selection. And what happens is that over time, more and more mutations accumulate, more driver mutations accumulate, and you get cell clones appearing with more and more aggressive growth phenotypes, and eventually metastases. So some key concepts to take away from this. Firstly, is that the passenger mutations far outnumber the driver mutations and are very numerous. Secondly, that these driver mutations are under positive evolutionary selection. And thirdly, that the genes or genomic elements in which these driver mutations fall are defined as driver genes, and these are key therapeutic targets. And so this has been known for a long time in the protein coding world, and it's quite standard theory, but in the world of long non-coding RNAs, there are no cases so far demonstrated of cancer driver link RNAs. And so with Andres, what we've been thinking about the last couple of years is whether you could have examples where you have a link RNA with a mutation, where the mutation somehow alters the function encoded in that RNA molecule and can confer this growth advantage onto the cells. And so, as I told you, this question was impossible to answer until recently because we lacked the data until this PCOG project, which was mentioned earlier, this pan-cancer analysis of whole genomes project, which is organized by the International Cancer Genome Consortium. So this is the first effort to comprehensively collect entire tumor genomes and, importantly, to collect the mutations in those genomes, to catalog thousands of maps of tumor mutations. And so this was a worldwide effort, more than 1,000 researchers across numerous different countries, 37 different tumor types, for a total of approximately 2,500 entire tumor genomes. And essentially, this data all boils down to a very simple format, although it's an extremely complex to generate this data. Overall, what you're looking at is 2,500 individuals and maps of the individual nucleotides which are mutated in them. So altogether in our data set, we have 45 million of these single nucleotide variants, or SNVs. And now the question becomes, if you're faced with this data set, how can you use this data to identify link RNAs or other elements which are driving tumors? And so this brings us back to the aeroplane. So the first, I'm going to show you two different philosophies and methodologies you can use to identify a driver element. The first one is pretty self-explanatory. This is mutational burden. So this is essentially saying with our aeroplane, now instead of studying one crashed airplane, you're studying 2,500 crashed airplanes. You start to chart where the damaged areas of this plane is, and then eventually the regions of the plane which are recurrently damaged in every crash, you can begin to infer are the regions which are causing the crash and not a consequence of it. And so similarly, we take our maps of link RNAs from standard annotations, we map all the mutations to these genes represented by stars, and we ask whether we see an enrichment of mutations in the functional regions of the link RNAs. And here we just assume very simply that exons, because we assume that the spliced RNA is functional, we assume that the driver mutations can only fall into exons, and that mutation rates in the background region represented by introns and surrounding regions are representing the background mutation rate. And so we're looking for genes now which are enriched in mutations in their exons over their local region. The second approach is slightly more complex for finding driver genes. This is called functional impact. Here, instead of looking at the number of mutations, we're actually looking at the nature of the mutations and specifically assigning a score for how or damaging or function altering we predict every mutation to be. And what we're looking for now is link RNAs not with increased numbers of mutations, but rather with mutations which are predicted to strongly alter the functionality of that encoded link RNA. This is much more challenging because actually we have a very poor understanding of the sequence function code of link RNAs. This is a big challenge. In our case, we use a standardized data set of base level predictions, which are quite widely used and actually give us rather good results. These are based on a combination of evolutionary conservation and other features. So Anders wrote this software called Exonator 2. The first Exonator was used in these PCOG publications, which I'll tell you about at the end. Exonator 2 is the first approach we've got, which actually integrates both of these different approaches, both the recurrence and the functional impact. In each case, it simulates the background expectation using intelligent randomization processes to simulate the mutational processes. And for each of these, it calculates a p-value. It combines these p-values together to get a gene-level significance value for being a driver for every link RNA in your data set. So now I'm going to show you. So then we developed Exonator 2, and then we went back and ran it on these 2,500 tumors from the PEACOCK project, looking for putative cancer driver lincRNAs. So that's what I'm going to show you here. On the x-axis, you've got different lincRNAs. On the y-axis, you have different tumor cohorts. So it's important to understand that in the PEACOCK project, we do analyses which we call pan-cancer analyses, where we just merge all of the different tumor types together into one big analysis to maximize our statistical power. But then we also separate the tumors into different groups based on the different cancer types. So you have less tumors, but they're more homogeneous. Okay, so that's what I'm showing you here. At the top, you've got pan-cancer, and then you've got all these different classes down here. So here's a summary of the findings. So firstly, there are 11 known cancer lincRNAs. So this is back from our manual annotation, which we did previously of known cancer-causing lincRNAs. Every time you see a colored cell, that's a significant hit for that lincRNA in that tumor. So we have many hits in the pan-cancer because it's usually more sensitive, but we have also different hits in different tumor types. The color indicates the fold enrichment of mutations over what you expect by random chance, and the numbers inside indicate the number of mutations that gene has got. So in addition to these 11 previously known cancer lincRNAs, we found 16 novel ones, and these were not previously associated with cancer. And you see that these are associated, for example, with head and neck, with prostate, and with liver. And we're following, actually, at this moment, following up these ones in the lab. So, of course, we could just be predicting random garbage. We have to know and have some confidence in the predictions of this program. And I'll show you, firstly, one thing which we're quite happy about is if you just take these candidate genes we discovered versus all the ones which are negative in our screen, and we look at how many of them were previously associated with cancer, this 11 known lincRNAs is a big enrichment over what you would expect by random chance. So even though Exonator is given no information about prior literature, it gives you back a lot of known previously studied cancer lincRNAs. So that gives us some confidence we're predicting something interesting. We've also now done a much more comprehensive analysis across all kinds of different features, which I don't have time to tell you. I'll just tell you about a couple, evolutionary conservation and expression. So now we're asking, do the lincRNAs we predict have signatures of being functional and important genes? So here you're looking at base level evolutionary conservation of these lincRNAs in their exons, and the blue ones are the candidates, the gray ones are all the other ones. We see that there's a significantly higher conservation on the nucleotides in the exons of these lincRNAs. Similarly, if we look at their expression in normal healthy human tissues, the predicted candidates are more highly expressed across normal tissues than the non-candidates. Also, just to summarize, they have all these other interesting features. They tend to be highly expressed in tumors. They tend to be differentially expressed in tumors, comparing tumor to normal. They come out more frequently than expected by chance in vitro functional CRISPR-I screens. They're also enriched for germline cancer SNPs, so these are predisposition SNPs for developing cancer, and also they're enriched for 2 other databases of cancer lincRNAs. So this is really quite a non-random set of lincRNAs which we're discovering using this program. And so obviously a very obvious question to ask then is, I've showed you this mouse prediction of cancer driver lincRNAs, I've showed you these human predictions, and do they agree with each other? And of course I wouldn't show you this unless they did agree with each other. There's a rather light but very significant overlap between these 2 methods. So using these 2 completely different methods, they're both converging on, at least partially, on a core set of lincRNAs which seem to be capable of acting as drivers in cancer in both human and in mouse. Okay, so now for the last part, this is rather controversial to claim that that lincRNAs can act as drivers, and indeed, if you go and look quite closely at these PEACOCK papers which have come out, one of the main take-home messages from these papers is that they find little evidence for lincRNA drivers, and so we need to have a much higher level of evidence to start claiming this. And Andres went into the lab, or he didn't go to the lab, but he talked to a colleague in the lab, Roberta Esposito, and with Roberta we came up with a strategy to try to answer this question directly in the lab. Can mutations in lincRNAs give rise to tumors or tumor-like phenotypes? So we took one of the candidates from Exonator. Here's the profile of the mutations across the 2,500 tumors. Here's a track of a nucleotide conservation. And manually, Andres and Roberta went through and found 5 regions of this lincRNA, which looked particularly interesting in terms of evolutionary conservation, density of mutations, and other processing features. So they chose 5 candidate regions, and what Roberta did is she designed a series of guide RNAs with wild-type Cas9 single guide RNAs to introduce mutations into this gene. And then Taisi and Paulina set up a very convenient assay in the lab, which we call the red-green assay. We introduce our mutations one by one using CRISPR into M-cherry, so red-labeled cells. We've got control cells carrying non-targeting guide RNAs. We mix these in equal proportions, and we ask how the color of the mixture changes over time. So if your mutation is a driver mutation, you expect the mixture to become more red. If it's a deleterious mutation, you expect the mixture to become more green. And if it does doing nothing, then the color should stay the same. So Roberta did a couple of controls. She targeted 2 intergenic genomic loci individually, and she saw no change in the proliferation of the cells. She knocked out this lincRNA, so actually this is known to promote growth of cells, and when you knock it out, you get a light but significant reduction in the cell growth. Now she started introducing her mutations into this candidate gene, and in at least a couple of these cases, we saw no significant difference in the cell proliferation. However, it's very slow. In several cases now, Roberta found that introducing small indels using this CRISPR, and these are 3 different locations, gave repeatable increases in growth to the cells where the mutations took place. So this is the opposite. This is not a loss of function, because we've shown loss of function causes less proliferation. This is a gain of function in this lincRNA at multiple locations. Also, that was done in HeLa, but also we repeated this in a completely different cancer cell line, and we saw the same effect in the control, no change in proliferation. Knockout, you see a loss of proliferation, and in several of these targeted regions, we also see an increase in proliferation, which is consistent with these mutations being able to confer an increased proliferative rate and possibly being driver mutations in early tumors. Okay, so I'm going to wrap up. So I showed you how we use mouse mutations to identify candidate driver genes in human. I showed you how we use human mutations to identify candidate driver genes. And I showed you some experimental evidence that these mutations can, in fact, alter the phenotype of cells and confer on them a growth advantage. I'll just show you one. So this is the same stuff that Ant and I showed you earlier on. We've been involved quite heavily in this PCOG project. If you're interested, Exonator, the first Exonator was part of this driver analysis effort, but we also have a paper where we cataloged very carefully a very good quality data set of cancer lincRNAs, and we also have a new version, which is, this was 122 examples, but now our new version is going to have 472. If anybody needs it, I'll be happy to share it if you email me, and you can find a lot of information from this whole project right here. So I just want to thank the team. I've told you the guys who did all the work were funded by these organizations. We're moving to Dublin, and we have some positions if anybody is interested to join. And thank you very much for your attention.